This is my genetics exam 4 prompt 2 mini exam video about prokaryotic transcription. So it has to start somewhere, and the place where transcription starts is called a promoter region. -10 and -35, those boxes are both promoters, and they help, the sigma helps find where to start the segment in the hollow enzyme. After the DNA is actually transcripted and made, DNA polymerase 1 and 3 go back and proofread them to make sure everything's right with exonuclease activity. DNA polymerase 3 can also catalyze the formation of phosphodiester bonds, which are the backbones that link DNA and RNA together along the sides. Whenever they're done, whenever the gene is done being transcribed, it has to end somewhere. So one of the ways that it ends is with a hairpin loop, which is when the mRNA is mirrored and signals back on each other, on itself. So it makes a little hairpin loop that stops the formation of more RNA. There's also a poly-A tail that's added to the end of RNA, and it helps prevent degradation of the molecule and makes it more stable. And then there's coding and template strands. So the coding strand has the same sequence as RNA, but the coding strand has T's and the RNA uses U's. And then the template is the template for the complementary RNA transcript. And back with the -10 and -35 promoters, since that is how the gene knows to start transcribing, if you don't have that, if it's mutated, then it can cause the gene to no longer start or to have issues with that, and then it can have reading frame errors or other things like that. Like, it's more prone to mutations or just not being made at all. That is my video.